I'm going to structure my talk in 3 quick pieces. The first part, just sort of an introduction to frame some of the questions, and then I'm going to summarize about 20 years of work that I've done in 10 minutes, focusing on one particular structure, the amygdala, that implements 2 particular mechanisms by which we find out what other people's internal states are. One sort of at the front end that involves directing our attention to salient features of faces, and one more on the reactive end that involves emotions that are triggered that in turn guide our social behavior. And then what I'll spend the last 5 minutes or so doing is expanding this out and asking what is it that we can do, not just with these rare cases that have lesions of the amygdala, we have 4 of them right now, but with hundreds of patients that have lesions all over their brain, what kinds of structure-function mapping can we extract from that? So the first question, if we step back very broadly and just think about all the different ways that we have of knowing about the world, you could imagine partitioning this very broadly into 3 categories. And these are not different kinds of things ontologically, but they're different ways in which we have access to the world. One, which we understand quite well how the brain does it, is knowledge of the non-social environment, tables and chairs, laser pointers that don't work well on the screen, and so forth. So sensory physiology tells us a lot about that. How does the visual brain process objects in the environment? There's a second source of knowledge that's much more mysterious and that I won't be talking about, which is how do we know what's going on inside our own minds, often apparently without the kind of sensory evidence that we have about the external world. How does that work? And then the third one, which is a big theme of this meeting and the only one that I will be talking about, is knowledge of what goes on in the minds of other people. And you've heard something about this already, and you might imagine very naively that perhaps 1 + 2 = 3. So all the evidence that we have to go on to figure out what's in the minds of other people depends on observing their behavior, their facial expressions, their gestures, and so forth. But then there are other mechanisms that are highly inferential, such that we can interpret what we see in other people by putting ourselves into their shoes, for instance. So one hallmark of social cognition, of figuring out what goes on inside other people's minds, is that it is intensely inferential. You might think that that is unique to social cognition. It's not. All of cognition is inferential. And as this visual illusion here illustrates, this is the famous Canizza triangle, what you'll see here, instead of these 3 corners and 3 Pac-Men, is a white triangle occluding an upside-down triangle in 3 circles. And the reason that you see this is that your brain interprets this as the most parsimonious explanation. And in fact, people have recorded in monkeys, you can find neurons in the visual system that will respond to an illusory edge up here. So there's no local contrast difference, but these neurons respond as if there were an edge there because that's the way that this whole thing is interpreted. So the brain does this all the time. It makes inferences about a spatio-temporal context, about the surrounding context in space in which a stimulus occurs and the context in time, namely memory. The same kind of thing, but in different ways, happens when we try and figure out what goes on inside the mind of another person. And again, it's very automatic, very ubiquitous, very effortless in most cases, and so it's easy to underappreciate how complex this is until you actually start building artificial systems to try and do it. But all you have to go on is what hits your retina in this case. So you see these contrast differences. Your brain interprets this as eyes and nose and mouth and features. But it doesn't stop there. It binds this into a holistic perception of a face. It doesn't stop there either, but it sees this as a person behind the face. So when we're looking at each other, when I'm looking out at the room, it's impossible to see faces just as a bunch of visual stimuli, but you automatically see them as people with intentions, emotions, and feelings, thoughts behind the face. And the question is, what are the varied mechanisms by which we do that? And they're varied. There are many of them. I will tell you just about 2 that we've looked at in my lab in relation to one particular brain structure, the amygdala, which is Greek for almond. The amygdala is a structure that's about a cubic centimeter or so, a little larger than that, one on the left, one on the right side of the brain, just sort of if you go in from your ears, about 12 million neurons per side. And it's been studied extensively in both animals and more recently in humans. The sort of workhorse task that people have looked at are forms of emotional learning, such as Pavlovian fear conditioning. And you can do experiments where what you do is you take a rat or a mouse and you pair some aversive stimulus like electric shock with a tone. And if you do that a bunch of times, the rat learns that the tone predicts the shock and behaves as though it is afraid of the tone. The amygdala is necessary for that type and many other types of emotional learning. And you can show this if you measure various indices of fear or anxiety. So for instance, if you measure respiratory changes, EEG arousal, increased startle, facial expressions of fear, all the varied components that would constitute fear, a behavioral state of fear or anxiety, can be triggered by the amygdala because it projects to many, many different structures, all of these places in the brainstem and hypothalamus, that each implement one of the components of fear. So what we'd want to do in social interactions and eliciting emotions is to trigger a very multimodal but coordinated set of cognitive and behavioral changes. The amygdala is able to do that because it projects to so many different structures that implement all of these different components. In fact, these are data from Malcolm Young in the cat, where they plotted the connectivity between the amygdala and many other brain structures. That's what all these different things here are. And the amygdala always ends up in the middle here in this sort of multidimensional scaling representation because it is one of the most densely connected structures in the brain. So again, just emphasizing the fact that the structure of the amygdala, the connectivity of the amygdala, to some extent reflects its function, which is to affect very coordinated but very high-dimensional changes in cognition and behavior. So it modulates attention and memory and decision-making and so forth, all based on the emotional or social meaning or the value of a stimulus. Not only is the amygdala embedded in a network in the brain like this, but recently people have looked in relation to social networks. I just wanted to show you one piece of data here. There are now several studies of this, both in humans and also in monkeys. With these people, this is work by Lisa Feldman-Barrett and colleagues at Boston University. What they did was take a bunch of people and quantify the extent of their social networks, their interactions with other people, which is what's plotted on the y-axis over here on the left. And they correlated that with the volume of the amygdala. And they found that there was a significant association such that the larger your amygdala, the larger your social network. People have now found similar relationships also for the number of friends you have on Facebook, and they have found this kind of relationship also in monkeys where they had more experimental control. So what might you imagine doing if you're interested in the functions of the amygdala in emotion and social behavior? And one of the easiest experiments you might imagine doing is to ask the question, what happens if you lesion the amygdala? So we've been studying a patient going by the initials SM and several others like her for a number of years and done exactly that. And I wanted to show you two quick pieces of data that illustrate the role of the amygdala in two particular mechanisms that come into play when you interact with other people. One mechanism where we teamed up with these people, Fred Gosling and Philippe Schins, they invented a method called bubbles, the details of which I won't go into, but essentially it depends on showing you little randomly sampled pieces of faces in order to extract from that what information from faces you use to make a certain kind of judgment, in this case, judgments of emotions. And what you get from that are pictures like this. So what this shows, these are statistically thresholded images that show you the region of the face where there is a statistically significant association between that part of the face and your ability to discriminate that particular emotion. So it makes a lot of sense. So you make use of the mouth in order to recognize happiness, use of wide eyes to recognize fear, a furrowed brow to recognize sadness, and so on and so forth. So we did this experiment. So these are the data. This is the result that you get from healthy individuals in a task where we asked people to distinguish between fear and happiness in our particular experiment. The eyes are very important in faces. People make a lot of use of eyes and faces in order to figure out how others feel, and to some extent also the mouth and the nose. When we did the same experiment with the patient who has amygdala lesions, we found a different classification image, and in fact, to show the difference between these two, you can subtract these. And again, these are all statistically thresholded results. So what this shows, this difference here, is this is the information from faces that you would use in order to judge the emotion, in particular fear, but you also use it for other emotions, that the patient with amygdala lesions fails to use. So it's a very specific deficit. We began with findings in the early 90s that showed that the amygdala was necessary to make social judgments about other people. So these patients with amygdala lesions are bad at making various social judgments from faces. They're particularly bad at recognizing fear in faces. And it seems to result from a very basic processing impairment that has nothing to do with emotions per se. So lesions of the amygdala abolish the ability to search out in the environment and pick up social information that is relevant or important in the first place. This patient with amygdala lesions and others like her also fails to direct her gaze towards the eyes and faces and doesn't make normal eye contact. So this is one mechanism in which the amygdala is involved sort of at the front end. It's very important. So right now, if I look at the room, it's not that I can just open my eyes and I get all this social information in and then I start processing. There's too much social information, and I need to have mechanisms in order to know where to look and how to explore the social environment in the first place. And the amygdala implements that function as one of its functions. There's a second function that is less instrumental and more reactive, and that has to do with this particular experiment that I'll show you here. And I was going to start by showing an experiment done by researchers in Korea, not in humans, but in rats, but it illustrates the way that you would measure emotional or social behaviors in rats. Well, you would put a video camera up, and you could, just like the view that we have here, you could quantify various kinds of behaviors of the animal as it moves around. And what they did in this particular study here is confront the rat with a frightening stimulus, this RoboGator. So here's a food pellet. This rat is enticed to go out and approach the food pellet, but there's this potentially dangerous object there, and you'll see how the rat behaves in just a second. And you can imagine quantifying various aspects of this behavior. So you can quantify the startle movement of the rat. You can quantify the latency that it takes to come out. So it gets longer and longer. This goes on for quite a while, and the rat needs to muster up more and more courage in subsequent trials to be able to come out of there. The reason I'm showing this to you is that what these investigators did was to make very precise pharmacological lesions then of the amygdala and ask the question, what happens to behavior if you lesion selectively the amygdala? And that's what's shown in this video here. So here again we have the same setup. We've made it even more difficult for the poor rat by putting the food pellet way over by the RoboGator. And this rat now has amygdala lesions. And I'll just stop for a sec so you can just see what happens. There's no response at all to the RoboGator. So this illustrates several points sort of very nicely and visually that also apply to our patient with amygdala lesions. Just like our patient with amygdala lesions, it's not the case that the rat has a basic sensory problem. It can see things just fine. It can move around. It's also not the case that there's a complete lack of any motivated behavior. It's motivated to seek the food pellet, but there's a very selective impairment in any kind of fear-like responses to a potentially threatening stimulus. So we did an experiment very similar to this in humans. Maybe I'll just demonstrate it with Jack. Here's the experiment.
I'm scared.
You're going to be my... You will be. Don't worry. I'm the experimenter. You're the subject. Stand up. Here's the task. It's very simple. You can do this at home. Ideal experiment. Very simple, large effect size. Here's the instruction to you. Walk towards me. Okay, so he has an amygdala lesion. Normally...
I wasn't sure what I was supposed to do then. Oh.
Normally, people stop at some point.
You picked the wrong guy.
The last
and you can.
you gave him was walk towards you.
That's what we tell participants in our study too. But
I noticed you were glaring at me at the end there.
We're just two good friends. But you can. So normally, people stop at some point, and you can actually do this under a variety with many different experimenters, and you can have the participant walking towards the experimenter walking towards the participant, and the participant has to say stop when he wants the experimenter to stop. And then you can quantify, just like you can measure the behavior of the rat, you can just simply quantify distance between people. Very simple experiment with fairly reliable results. What you get is something that looks like this. So here on the x-axis, we're plotting interpersonal distance in meters, the experimenter standing over here, and these are a bunch of healthy individuals. So some people like to stand far away, some people stand closer, but everybody except Jack stops at about 0.4 meters or so because you bump into this wall of personal space. And if you get closer than that, it starts to feel very uncomfortable, and so people stop. Whereas the patient with amygdala lesions didn't. So in this experiment, in fact, this just shows the mean interpersonal distance that she preferred. In several trials, she walked all the way up to the experimenter, just like Jack did, to touch the person's nose. And there was no, on questionnaires and also psychophysiologically, there was no strong emotional reaction to invading another person's personal space. So these are two mechanisms by which the amygdala contributes to social behavior. One is at the front end, a more instrumental role for figuring out what's salient and important in the environment. Another example, and this is just one example of that mechanism, is that it elicits emotions that in turn guide our social behavior, as in this particular example. The last 5 minutes, So most of this work was done in that one subject. We now have 4 others like her. They're extremely rare because getting selective bilateral lesions, you would never get that kind of damage from a stroke. These lesions are due to a very rare genetic disease that these patients have. But of course, you can scale this up and ask, what can we glean from large-scale lesion mapping? And this has an interesting and important history. So as you probably know, Franz Josef Gall and his followers in the late 1700s, early 1800s started phrenology, and the whole idea was that there were particular functions localized in particular parts of the brain. We can deride this nowadays. People thought that you could figure out the extent to which somebody had this function by palpating their skull and seeing bumps on the skull, etc. But of course, they were right in one respect, which is that the brain is not equipotential. Different parts of the brain do different things. They were wrong in 2 very interesting ways that are worth thinking about. One is that no cognitive function is localized in one structure, but rather we should think about distributed networks. So coming back to the amygdala by itself does absolutely nothing. It functions as one component of a distributed network, going back to the picture I showed you at the beginning. And so the task is to understand, how does the amygdala function in that network? What is the network for mediating, figuring out that eyes are important or mediating emotional responses to a person? There's a second respect in which they were wrong, but in interesting ways, which is, what are the cognitive functions to map in the first place? And so they had a list, like this one here. Some of these might make some sense to us. Many of them seem very silly, like a poetical talent presumably isn't localized in any way or has no dedicated brain system. But the point, I think, sticks even nowadays. The question is, what are the functions to map? And we have attention and memory and decision-making and, of course, much more fine-grained sort of subdivisions within those. Are those the right way to carve up the mind, or are there other kinds of categories that we should be trying to map? Well, the mapping that I've been doing has been with my colleagues ever since I was a postdoc with Antonio Damasio, drawing on the very large lesion registry that's there at the University of Iowa. This just gives you a few facts about it. But there are available several hundred subjects that you can now map. And so you can take a bunch of patients and you know the etiology of their disease and lots of other information about them. And if you just ask, so in this particular study there were 240 patients, where are their lesions, you can sample most of the brain. There's some regions of the brain that are not sampled. There's some regions that are sampled more densely than others. These colors here show the density of overlaps of lesions in these 240 patients. So a lot of lesions had overlap in damage in these regions here, because those are regions where you would commonly have stroke, for instance. But then what you can do is you can test these 240 patients on all kinds of cognitive tasks and ask, where is there a systematic association between having damage in a certain place and impairment in a certain task? So this gives you a kind of causal level of understanding that neither fMRI nor electrophysiology does. It shows you which regions of the brain are not just sufficient for or involved in some function, but necessary for it, in the sense that if you lesion that region, you impair that particular function. I'm not going to go into the details here, but just to give you a flavor of the kinds of pictures that you can get, if you have a sufficiently large sample size and if you have a sufficiently homogeneous sampling of lesions all over the brain, you can do voxel-based lesion symptom mapping, where you can now generate statistical maps corrected for all the different places that you sample all over the brain that show you where is there a statistically significant association between having damage in that place and impairment in that task. In this particular example, these were abilities extracted from the Wechsler Adult Intelligence Scale. This is the ability to visually organize things. This is verbal comprehension, and they localize to regions that make a lot of sense. You can go further than that, and you can abstract much more abstract kinds of cognitive functions. So in another study, for instance, what we did was to take all of the individual tasks of which the Wechsler Adult Intelligence Scale is comprised, and take out of that these basic factors like verbal and spatial and working memory function, and ask, what is the residual variance left that is shared across everything? And that is a factor called Spearman's G, which is sort of the idea is it's fluid general intelligence that is not specific to any domain, but is maximally shared in variance across all the different tasks. And you can map that. And when you map that, you get a picture that probably makes a lot of sense once you start thinking about it. There are regions of the brain where general intelligence seems to depend on, in the sense that if you damage that region, you impair general intelligence. But there are many of them, And many of these regions actually involve white matter connecting regions rather than cortex. Coming back to the picture that eventually what we need to understand is not single brain structures, but brain networks. So to finish up in terms of future directions, each of us could list 20 interesting future directions. I will restrict myself just to sort of this list here pertaining to lesions. What historically has been there, you saw Broca's brain earlier, there's been historical case studies, and you can get a lot of information, just like we did in this patient with amygdala lesions, from one case. Very informative. That's been now expanded to larger groups. You can go up to several hundred, as in this patient registry from the University of Iowa, but there are no big consortia that provide any kind of infrastructure. There's lots of work being put into big databases, big repositories for fMRI information, but if somebody here has an idea and wants to test this in a lesion way, it's very difficult. So one thing I think that would be extremely important to set up is some large consortia. So you would have thousands of patients that have been well characterized with lesions, and then that can spin off various kinds of questions about mapping particular cognitive functions in those patients. And then the second one is something that we are now doing and others are as well, which is, of course, to combine lesions and fMRI in the same patients. There's no reason that you can't take a patient with a lesion and also do an fMRI study. So you could take this patient with amygdala lesions, put them in an fMRI scanner, and ask what is now the difference in activation of all those intact brain structures that would normally receive input from the amygdala that's no longer there. And that would give you insight into the sort of causal role that the amygdala plays as one component of this big network. So let me finish by acknowledging the funding sources that have made this work possible and the many people, not all of whom are listed in my lab, and collaborators elsewhere. Thank you.
Thank you.